we set out with Sergio Baronzini and Steven Saucer on behalf of the International MS Genetics Consortium to perform the largest genetic study of MS severity to date, and we sought to answer the following questions. Are there genetic contributions to MS severity? So far, none had been found. Can we link genetic variation to diverse and heterogeneous patient outcomes that we all observe? Can genetics help inform our understanding of mechanisms underlying MS severity? So to address this, we assembled 15,000 people with MS with both clinical and genetic data, and following quality control, 12,500 individuals were studied. We then replicated our findings in an independent cohort of 10,000 people with MS. All these individuals were whole genome genotyped, and we had, after imputation, about 8 million genetic variants to analyze. So we did not just include any person with MS, we preferentially recruited individuals that were older and had longer disease duration, so on average aged 52 years and with a 20-year disease duration, with the notion that these individuals will have effectively declared their long-term outcome. Disability was measured pragmatically using the EDSS and then converted to the age-related MS severity score by ranking disability against individuals within a 2-year age strata. We also had access to longitudinal data that allowed us to do additional analyses that I'll show in a few slides. So to the results, we identified the first genome-wide significant variant associated with MS severity, as well as an additional 11 variants that we brought forward for replication in that independent cohort of 10,000 people. 2 of these variants, including the genome-wide significant variant, successfully replicated, and not only that, but showed consistent effect size and direction of effect across the 9 cohorts that composed that replication data set. Now, it's worth noting that these 2 variants had not been previously reported and are not associated with the risk of developing MS. We then turned to longitudinal analyses using a subset of 8,300 participants evaluated over 54,000 visits and spanning up to 14 years. And here we found that individuals that carried 1 in red or 2 copies of the risk allele experienced faster 24-week confirmed disability worsening. Homozygous carriers also experienced a 3.7-year shorter time to median EDSS-6, so to requiring a walking aid. And in linear mixed model analyses, including all 54,000 study visits, you can see that individuals that carry this risk allele also accrue disability significantly faster. The same pattern of significant association across all of these outcomes was observed for that second variant, which was low frequency. So here we analyzed it just as carriers versus non-carriers. And here carriers had a 2.2-year shorter time to EDSS-6 or to using a walking aid. So having established these associations in the clinical data, we then turned to pathology to start understanding whether we can see associations with features that may be suggestive of progressive disease. And so to do that, we partnered with our colleagues at the Netherlands Brain Bank, and we started by looking at rates of cortical demyelination and brainstem lesion load, which we thought were important. And we saw that, again, homozygous carriers of this risk variant had a 2-fold higher rate of cortical demyelination and a 2-fold greater brainstem lesion count. And in an extension of this work, Yin Engelberg, also at Amsterdam, has been able to show that these homozygous carriers also have higher microglial activation, as evidenced by a higher proportion of foamy macrophages. Microglia. They also had increased evidence of axonal stress based on the presence of EPP-positive bulbs in the pyramidal tract. And they also had reduced neuronal density in the middle frontal gyrus, showing, again, broad associations with features that we think are relevant to the biology and pathology of progressive disease. Now, turning from clinical and pathological data to imaging data, our colleagues at TUM, led by Christiane and Mark, also showed that this risk variant accelerates the rates of brain atrophy by 28% yearly. And these are findings that were replicated exactly by the group of Ingrid Kokum at the Karolinska Institute. Having now established these different associations spanning clinical imaging and pathology, we wanted to see if we could start understanding the biology of what this variant is doing. And so to do that, we first tried to prioritize genes that are likely being impacted by that variant, and for that we used a range of sources of evidence that I don't have time to get into, but we found shared evidence for an effect on two genes, the first being dysferlin and the second ZNF, zinc finger protein. So dysferlin, some of you may know it as a protein that is that can be mutated in the setting of muscular dystrophies, but it is a protein that is present across pretty much all cell types, and that's really important in membrane repair. And then this second zinc finger protein, ZNF638, also called NP220, is a transcription factor that is important in repressing unintegrated viral DNA. This is work that's been shown in a recent Nature paper, and this was work done exclusively on retroviruses. So we don't know yet whether this is also important for viruses like EBV, but this is work that's currently ongoing. And it may also almost certainly be important in repressing endogenous retroviruses, which we think are also important in MS. Interestingly, this same gene, ZNF638, was also identified independently using single-cell RNA sequencing in a recent study by the Karolinska group that showed that not only was this gene differentially expressed across lesion types in MS, but was also enriched in a subset of oligodendrocytes that they called OLIG5, not that the number matters, but that was important in the membrane assembly and remyelination. So we next wanted to understand where these genes are being expressed. So we looked first at tissues and saw that these two genes that I've talked about so far, as well as the third gene that was prioritized from that second low-frequency variant, were expressed in the brain. And using single-cell RNA sequencing data, we were also able to show that they have high expression in oligodendrocytes and oligodendrocyte precursor cells with shared cell type specificity for oligodendrocyte lineage cells. That's at the RNA level. We wanted to prove this at the protein level. And so what you're seeing here are again stains from brains from people with MS, showing that ZNF638 and dysferlin are indeed highly expressed or translated into proteins as well in oligodendrocytes. And in fact, their levels correlate with the presence of oligodendrocytes. So we've talked so far about a single locus and what it might mean. Now let's take a step back and look at the full genome-wide data. We know that buried in the distribution are a lot of variants that will be important for MS severity, but that we just don't have the power to identify yet. And so when we sum up the overall genetic contributions to MS severity, we can find that at least 13% of MS severity, as defined here by the age-related MS severity score, is due to genetic factors. Now, this is just common variants. I've talked at the beginning about rare variants. We are not able to capture those yet in this data set. And so this number is certainly an underestimation, but it's already important. We next asked if we could identify enrichment for that heritability in particular tissues. Now, this takes a few steps that I'll quickly take you through, but we started with gene expression data from 205 tissues from all over the body. We then identified the genes that were most specifically expressed in a given tissue, and then used those genes to annotate the genome and then repeat our prior analyses on trying to ask, are there specific contributions from genes expressed in that tissue to MS severity? And when we apply this to MS susceptibility first, we see a strong enrichment in lymphoid organs and immune cells, which is what one would expect. But when we apply this to MS severity, you see a totally different pattern here with significant enrichment exclusively in CNS tissues, including multiple brain regions and the C1 segment of the cervical spinal cord, indicating that genes that are most specifically expressed in the brain are those that are most relevant to MS severity. We then formally compared the genetics of MS susceptibility and severity, and we can see that there is little overlap, with the correlation coefficient being just 0.17 and not even significant. We then wanted to understand if we could use this genetic data to identify risk factors for MS severity, so associations with other traits. And here we leveraged something called Mendelian randomization that I'll briefly run you through here. So Mendelian randomization uses genetic variants to proxy an exposure and determine its effect on an outcome. So to explain that, let me go through an example. Let's start with a gene called CYP2R1, which catalyzes the hydroxylation of vitamin D into its active form. We know that individuals that have an adenine at this position have, on average, higher vitamin D levels than those that carry a guanine. What we can do is then divide the population based on their genotype, and we know that they will differ on average with respect to their vitamin D levels. That's how we divided them, but not with respect to potential confounders. And then we can compare, in this case, MS susceptibility, but that's what we did for MS severity as well. And this mirrors, in some ways, a randomized controlled trial, except that the treatment and control assignments are due to genetics, to nature, rather than an experiment. And so what do we find when we apply that? Well, for vitamin D, we find nothing, a null effect, which is consistent with the recent clinical trial data showing that vitamin D supplementation does not affect MS outcomes, which is different from MS risk. And then when we look at body mass index, we see that there is a small positive association, but it is not statistically significant. We see that consistent with a wealth of observational results, lifetime smoking increases MS severity, and we see an interesting inverse association with years of education. So we wanted to understand that better. And we applied a series of both genetic but also epidemiological studies in the Swedish registries in GEMS and AIMS, and saw that indeed higher levels of educational attainment were associated with lower MS severity, again, both from the epidemiological data where we have realized educational attainment, as well as from the genetic data. And we can see that this remains significant after adjusting for socioeconomic factors such as income, smoking, and so on. And then we wanted to broaden our question, not looking specifically at causality, but just what other diseases might the genetics of MS severity look like? And so when we apply that to MS risk, we see that MS correlates strongly with other autoimmune diseases, very little with neurologic diseases. And this is a topic that I recently had the pleasure of reviewing with David Hafler, and that Matt Lincoln in the audience here has done a lot of work on. And interestingly, again, for MS severity, different from MS risk, we don't see any correlation with autoimmune disorders. We don't see any correlation with neurological disorders, but we see, again, this inverse association with general cognitive functioning as well as with aging phenotypes, both lifespan and health span. I'm going to skip over this section, which basically just says that we have not found any evidence of an effect of MS risk variance on severity, which is what I told you at the beginning. So to summarize, we studied 8 million genetic variants in over 22,000 people with MS and identified the first genome-wide significant and replicated modifier of MS severity. The lead variant conferred us a faster time to EDSS-6 by nearly 4 years, with a consistent effect on pathology and MRI data. We have also shown you that both individually and in aggregate, these genetic associations point to genes expressed specifically in CNS and neuronal and glial cells as being the most important in determining MS outcomes, which indicates to us that while MS seems from the genetic landscape to originate from a defect in the immune system, when we talk about who will do well or poorly, it is CNS resilience that seems to determine the outcome. And finally, we hope that this will serve as a starting point. So we're expanding on this study by including many, many more people with MS, reaching, we think, up to 80,000 people. And by starting to uncover these genetic effects on MS severity, we hope to be able to provide evidence for or against potential drug targets and support the community's push in treating progressive MS. Last word, all of the data that I've shown you so far included individuals from European ancestry. We know that it's important that we improve our inclusion of non-Europeans in these genetic studies, and we're also currently working on doing exactly that. With that, I'd like to thank Sergio Baronzini and Stephen Saucer for their mentorship, as well as all the members of the IMSGC and Multiple MS, and once again, thank MS Canada as well as the other funders. Thank you very much.